Welcome back for another deep dive. Today we've got a fascinating case study from the Journal of Comparative Pathology. It's about this, get this, an 18-month-old Cane Corso dog with an epithelioid glioblastoma. And you ready for this? It had metastasized to the lungs.
Wow, that's pretty young for a glioblastoma, isn't it? And lung metastasis, too. That's not something you see every day.
Yeah, definitely not. And that's why we're diving into this report today. We're going to break down the clinical presentation, the diagnostics, and the postmortem findings.
Sounds good. So where do we start? What brought this poor dog to the vet in the first place?
Well, it all started with some sudden neurological symptoms.
Okay, like what?
Ataxia, recumbency.
Uh-oh. That doesn't sound good.
No, it wasn't. And it progressed really quickly.
Yo.
Urinary incontinence, seizures, even nystagmus.
Oh, man. So what happened?
Sadly, the dog's condition just kept getting worse and the owners opted for euthanasia.
That's heartbreaking. I can only imagine how difficult that decision must have been. So what did they find during the postmortem?
Well, as you might expect, they found a large mass in the brain,
Where specifically?
left cerebral hemisphere. And it had all the classic signs of aggressive growth hemorrhage, necrosis, you name it. But here's the kicker. They also found a mass in the lung.
Wait, in the lung, as in singular?
Well, there was one main mass in the right caudal lung lobe, but there were also a bunch of smaller nodules scattered throughout both lungs.
So it wasn't just a brain tumor, it had spread.
Yep. Pretty unusual, right? Especially for a glioblastoma in a dog. So they did what any good pathologist would do. They took a closer look under the microscope.
Of course. The cells in
The
the...
brain and the lung lesions were basically identical.
Really? What did they look like?
Arranged in sheets and packets, very little supporting tissue, poorly differentiated, with a high mitotic index.
How high are we talking?
They counted 28 mitoses in 10 high-power fields. That's definitely indicative of a fast-growing tumor.
Absolutely. Anything else notable about the microscopic appearance?
Oh yeah, Serpentine Necrosis, hemorrhage, glomeruloid microvascular proliferation, all the hallmarks of a really nasty tumor. And to make matters worse, it had invaded the subarachnoid space.
Yikes. So how did they ultimately confirm it was an epithelioid glioblastoma? Those can be tough to differentiate from other brain tumors, right?
Right. They used immunohistochemistry, or IHC for short.
Makes sense. What markers did they use?
Well, they started with vimentin, GFAP, and Olig-2.
And the results?
Strong vimentis positivity, which pointed to a mesenchymal origin, variable GFE staining, suggesting an astrocytic tumor, and about 30% of the cells were positive for Olig-2.
Okay, so that narrows it down, but how did they definitively rule out other tumor types?
They also checked for markers of epithelial and neuroendocrine cells, but those were negative.
Gotcha. So all signs pointed to an epithelioid glioblastoma. Anything else interesting in the IHC panel?
Yes, they also stained for connexin 43.
Interesting. And
About 80% of the cells were strongly positive.
connexin 43, that's a gap junction protein, right? What's the significance of that in this case?
Well, in glioblastomas, connexin 43, or CON43, is linked to increased invasion and chemoresistance.
So its presence here could explain why this tumor was so aggressive and why it spread to the lungs.
That's one theory.
Fascinating.
Yeah.
There's definitely a lot to unpack here. It's wild to think about, an 18-month-old dog with this kind of tumor. You just don't see that very often.
It really is unusual. Do we know anything about why this type of tumor might show up in such a young dog?
Unfortunately, not really. We still don't have a great grasp on what causes glioblastoma in dogs in general.
So more research is needed.
Definitely, especially when it comes to cases like this one, where the dog is so young. There's got to be something going on there, whether it's genetic or environmental or who knows what.
Makes sense. Now, let's talk of the elephant in the room.
Yeah.
The lung metastasis. We know it's rare in canine glioblastomas, but in this case, it did happen. What makes the lung such a common target for metastases in general?
Oh, well, think of it this way. The lungs are basically a giant filter for the blood. They're constantly being bombarded with blood flow from all over the body.
Okay, that makes sense.
So if there are any tumor cells floating around in the bloodstream, the lungs are a prime spot for them to get trapped. It's all about opportunity.
So why wouldn't we see lung metastases more often in canine glioblastoma, given how the lungs work?
That is the burning question. And to be honest, we don't have a clear answer. It could be that the blood-brain barrier is doing its job a little too well, keeping those tumor cells locked up in the brain.
You mean preventing them from getting into the bloodstream in the first place?
Exactly. Or maybe it's just a matter of timing. These tumors are so aggressive that most dogs don't live long enough for the cancer to spread beyond the brain. They're euthanized before it has a chance to really take hold elsewhere.
So we might be missing a piece of the puzzle because we're not seeing the full picture of what these tumors can do.
Precisely. And of course, we can't rule out the possibility that some tumors are just inherently more likely to spread than others due to their specific genetic makeup or other factors.
Do we know if there's anything unique about epithelioid glioblastomas that might make them more prone to metastasis?
Now, that's a great question, and honestly, it's an area that needs more research. We know that in humans, epithelioid glioblastomas are pretty aggressive, so it wouldn't be surprising if they had some intrinsic biological features that made them better at spreading.
It seems like every answer just leads to more questions.
That's science for you. This case really highlights the need to dig deeper into the molecular drivers of metastasis in canine glioblastoma. We need to understand what makes some tumors spread and others stay put.
And what about the challenges researchers face when studying these tumors in dogs? What are some of the roadblocks?
Well, one of the biggest hurdles is simply the rarity of glioblastoma in dogs. It's not a common cancer, so getting enough samples for large-scale studies can be tough.
And I imagine translating findings from human research to dogs isn't always a smooth process.
Right. While human and canine cancers have some things in common, there are definitely differences too. What works in humans might not work the same way in dogs, and vice versa.
So we really need dedicated research specifically focused on canine glioblastoma.
Absolutely. If we want to develop effective treatments for dogs, we need to understand the unique molecular landscape of these tumors in their species.
It all comes back to the need for more research. Okay, so let's shift gears a bit and talk about the implications of this case for veterinary practice. What are the key takeaways for vets on the front lines?
I think the biggest lesson here is that vets need to be on the lookout for glioblastoma, even in younger dogs. We often associate it with older patients, but this case shows that it can happen at any age.
So don't rule it out just because the dog is young.
Exactly. Age shouldn't be the deciding factor. And while metastasis is rare in these cases, it's not impossible. Vets should be paying attention to any signs of illness that go beyond the neurological symptoms, just in case a cancer has spread to other parts of the body.
So don't just focus on the brain, consider the whole picture.
Right. And on a broader level, this case emphasizes the need for continued research in veterinary neuro-oncology. We've got a long way to go in terms of understanding these tumors, improving our diagnostic tools, and finding better treatments.
It's a call to action for the entire veterinary community. Now, I want to circle back to some specifics from the case report that stood out to me. The authors really emphasized the role of immunohistochemistry in diagnosing this tumor.
Yeah, they used it to rule out other possibilities and confirm that it was indeed an epithelioid glioblastoma.
Right. They looked for markers of epithelial cells and neuroendocrine cells, and those were negative.
Which helped them narrow down the possibilities. And the positive staining for Vimentin and GFAP, along with the OLID2 results, all pointed towards an astrocytic tumor.
And then there's that connexin 43 staining. We talked about it earlier, but it's definitely something that deserves more attention.
Agreed. Finding strong C/43 positivity in so many of the tumor cells is a pretty significant finding. It makes you wonder about its role in tumor invasion and metastasis in dogs.
Could it be a prognostic marker? Or even a target for future therapies?
That's the crux of the matter. It's certainly worth investigating further. This case really shows how much we can learn from even a single case study. It can spark all sorts of new research avenues.
The authors also brought up the limitations of our current prognostic markers for canine glioblastoma.
Yeah, like IDH1 and MGMT, those are used in human glioblastoma, but they haven't been fully validated in dogs yet.
Which just underscores the need for more canine-specific research.
Exactly. We need to figure out which molecular markers are truly reliable for predicting outcomes in dogs with glioblastoma. That's the key to making informed treatment decisions.
It's a complicated field, but cases like this help us move forward one step at a time. Now, there was one more thing in the report that I found pretty interesting.
I'm all ears.
The authors provided a really detailed description of the tumor's appearance under the microscope. They mentioned things like a high mitotic index, the presence of serpentine necrosis and hemorrhage, and glomeruloid microvascular proliferation.
All classic signs of an aggressive, fast-growing tumor.
And they also highlighted the invasion of the subarachnoid space. That means the tumor was spreading locally and had the potential to invade surrounding tissues.
That's a key observation. It shows just how insidious these tumors can be.
Even if you can remove the main tumor mass surgically, there's always a chance that some microscopic cells have already spread to nearby areas.
That's one of the reasons glioblastomas are so tough to treat. Their ability to infiltrate surrounding tissue makes complete removal extremely difficult.
So it often comes down to a multi-pronged approach. Surgery, radiation, chemo, all aimed at wiping out any lingering tumor cells.
Exactly. But unfortunately, even with all that, the prognosis for dogs with glioblastoma is still pretty guarded.
It's a stark reminder of the complexity of this disease and the urgent need for research to improve outcomes. Well, we've covered a lot of ground today, haven't we? This case study really takes you down the rabbit hole of epithelioid glioblastoma in dogs.
It sure does, and it highlights just how much we still don't know about this disease, especially when you consider how aggressive it can be and how rare extracranial metastasis is.
It's definitely a reminder for vets to keep an open mind, especially when dealing with younger patients. You can't rule out glioblastoma just because a dog isn't a senior.
Absolutely. And even though metastasis isn't common, it's not impossible either. Vets need to be on the lookout for any signs that something else might be going on, beyond just the neurological stuff.
Think outside the brain, so to speak.
Exactly. And this case also just underlines how important ongoing research is in this field. We need to know more, plain and simple. More about how these tumors work at the molecular level, how to diagnose them more accurately, and how to treat them more effectively.
It's a challenge to the whole veterinary community, really, to keep pushing the boundaries and finding better answers for our patients. But I think this case also raises some bigger questions that go beyond just this one dog and this one disease.
Oh, like what?
Well, we've been talking about canine glioblastoma, but what about human glioblastoma? Are there parallels? What can we learn by comparing the two?
No, that's the million-dollar question, isn't it? Could studying cases like this one in dogs actually lead to breakthroughs in human glioblastoma research? It's certainly possible.
I mean, maybe there are clues hidden in these canine cases that could point us towards new therapeutic targets or help us understand why some tumors are more aggressive than others.
It's fascinating to think about. It really shows you how interconnected human and animal health can be.
Exactly. This isn't just about dogs. It's about expanding our knowledge of cancer as a whole and finding ways to fight it more effectively, regardless of the species. It's about recognizing that the pursuit of knowledge and the search for better treatments can benefit everyone.
Well
Said. And on that note from Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.